For the sake of environment and marine life, whenever an oil spill occurs, an immediate response is needed in order to minimize or prevent damages. To do that, there are many existing approaches that have been used for oil spill cleanup, such as skimming, dispersion, and in-situ burning. However, dispersion via synthetic chemical dispersant is the most common one. Chemical dispersion ensures oil in water emulsions, in which oil is dispersed in the form of smaller droplets in water to enhance the biodegradability of oil. Within these synthetic chemical dispersants, there are many amphiphilic surfactants that manage to reduce interfacial tension between oil and water, which is necessary for the dispersion of oil in water. However, it has been reported in recent studies that synthetic chemical dispersants increase the toxicity of oil spills compared to natural or bio-based dispersants. Because of that, an alternative was made. Dispersants made from natural organic products have been reported to be more environmentally friendly. Proteins are used as emulsifiers because of their amphiphilic nature. They can be absorbed into the water interface, where they can distort the water and oil interfacial tension and reduce it. So to make our research even more environmentally friendly, the proteins we utilized were extracted from food waste, specifically from fish waste, which offers the good benefits of emulsification and environmental preservation. This is particularly important because this has been really considered in the case of crude oil dispersion.
As for the material, 3 fish waste samples were used. 2 samples are waste from a fish farm, and the 3rd sample comes from a lobster waste. Artificial seawater was prepared by dissolving artificial sea salt with an anti-caking agent in deionized water, and different concentrations of 10, 20, 30, 35, and 40 grams per liter were prepared. Lastly, the crude oil was collected from Zatco. In our research, we focused on 4 important methods. First, the preparation and characterization of protein isolate. Second, the preparation and characterization of the dispersants. Third, the oil and water dispersion. And fourth, the effects of neutral conditions using lecithin. For the preparation and characterization of protein isolates, the 3 fish waste samples were blended to ensure the size reduction. Protein isolates were extracted from the blended sample using an alkaline hydrolysis. The protein isolate samples P1, P2, and P3 were analyzed to confirm the presence of the protein functional groups in the sample using the FTIR spectrometer. A Raman spectra was also used to examine the significant differences in the amorphous structures of the protein isolates.
For the preparation of dispersant samples D1, D2, and D3, about 100 ml of glacial acetic acid was added to the protein isolate samples P1, P2, and P3. Then the suspension that was formed was mixed in an incubator, and the pH of each sample was adjusted using NaOH. Then the effectiveness of these dispersants was determined by measuring the emulsifying activity, and the stability of these dispersants was also investigated in terms of their ability to withstand gelling and forming. The ammonium nitrogen and the total nitrogen content of these dispersants were also determined as a measure of their protein content. As for the oil and water dispersion, artificial seawater of specified concentration was prepared by injecting a drop of crude oil into each sample. Each dispersant suspension that was prepared previously was also added to these samples in 1:1 volume ratio. The operating conditions employed for the preparation of the oil and water dispersion can be seen in the table, and the amount of oil dispersed was measured using a spectrophotometer.
For the effect of the neutral conditions, the solvent was changed from acetic acid to lecithin to study the effect of neutral conditions on the ability of P1 to disperse crude oil, because P1 showed the least ability for crude oil dispersion, as it will be further discussed in the result. It was prepared and characterized the same way as the other dispersants. Moving on to the results, the activity of each dispersant was first measured in order to determine the ability of the dispersants to form a homogeneous solution. These measurements indicate how well the protein samples were able to interact with crude oil. The particles of the protein isolate that did not form a homogeneous solution settled at the bottom of the dispersant. Through this, the 3rd dispersant sample was found to be the most active in the emulsifying activity, with a value of 94.5%. At room temperature, only 2 layers were observed in all dispersants, a lower serum layer, and the upper creamy layer. These layers were also observed after 30 days. The foaming capacities for all dispersant samples were measured. Since foam is unreactive, it reduces the activity of the protein. T3 exhibited the least foaming capacity of only 1% since it has a high emulsifying activity value. The dispersant samples were exposed to a high temperature during the gelling experiment. The protein isolate that was previously found as a precipitate mixed thoroughly this time. However, no gelling behavior was observed and the precipitate reappeared after the end of the gelling experiment. This indicates that the dispersants are stable and the continuous phase was preserved. Proteins with the higher amino acid levels often have higher solubility and activity. So the ammonium nitrogen concentration of the dispersants were utilized as a measure for the amino acid levels. The third dispersant sample showed the highest concentration, which confirms its relatively high solubility and emulsifying activity value. For the FTIR, the infrared absorption peaks that correspond to the aromatic amide and the amino functional groups in protein were observed in the IR spectra of the protein acids. Moving on to the Raman. The purity of the protein samples was also confirmed using the Raman spectroscopy, as shown in the figure. So there are no sharp peaks, which indicates the absence of crystalline impurities. This means that the amorphous structure of the protein were maintained and any unwanted materials were removed during the synthesis of the protein isolates.
So for our results, to measure the concentration of crude oil dispersed by the dispersants, the calibration curve of absorbance versus crude oil concentration was employed. It should be noted that D1 was sensitive to salinity changes. Crude oil dispersion by D1 began to decrease when the salinity was increased to 35 grams per liter and greater. Increasing the salinity decreases the solubility of dispersants in water, resulting in more dispersants being available to interact and mix with the oil, which makes it more effective, because surfactants cannot migrate to the water phase. This explains the slight changes in the dispersion effectiveness of D1 on different salinities until the value of 35 grams per liter. And at the very high salinity levels, salting in of the dispersant occurred and a decrease in dispersant effectiveness was observed. We also investigated the effect of the change of solvent from acetic acid to lecithin, which was observed. So the absorbance by D1 under different operating conditions were converted to dispersed crude oil concentration can be seen from this table. Dispersed concentration increased with increasing salinity. This is different from what we observed in results using acetic acid. So therefore we can attribute this to the effect of pH. Meanwhile, when the temperature was increased, the concentration of dispersed crude oil decreased until 45 Celsius, where there was no more dispersed oil. Meanwhile, the dispersed oil concentration increased with increase during time, and the dispersion even increased 6-fold when the stirring time doubled. And lastly, an increase in dispersed oil concentration was also observed when the stirring time was increased from 200 RPM to 250 RPM. In conclusion, oil dispersants can be created using protein isolated from fish proteins in both acidic and neutral pH conditions. Out of the 3 protein isolates we used in the study, the third sample, P3, proved to be the most suitable for crude oil dispersion because of its high dispersion activity, its large pore size, and it's considered very soluble as it was able to dissolve more in its dispersant suspension. In addition, it had the least foaming capacity, which made it more reactive and it enabled it to interact more with crude oil. Also, by measuring the levels of nitrogen, P3 had the highest amount of NH4 and nitrogen and total nitrogen contact, which correlated with higher dispersion activity. Lastly, this project is very important when it comes to the environmental aspect, as it provides alternative solutions and sustainable ones to current problems that are affecting our world. We would like to thank Dr. Hanifa Belushi, Dr. Giwa, and Ms. Bashar, and Khalifa University for providing us with the appropriate funding and the help we needed to conduct the experiments and to conduct the study. And we would like to thank ADU for providing us with this opportunity to showcase our skills.